Because I'm really a clinician, that treats patients, and I'm a scientist as well. I did a PhD at UCL in immunology, so you could call me a clinician scientist, but it's only in the last few years that I became quite interested in innovation and taking things to the bedside, because I realize unless you do that, you really don't get new technologies to the bedside. I think as academics, we often make the mistake of making seminal discoveries, but we publish them and then it's never taken forward. So really nobody benefits from them. So TB, as some of you would know, is a disease that's been with us for thousands of years. It co-evolved with humans for over the last 10,000 years. And there are many famous people who've had TB, Mandela, Gandhi, various other people. And what TB is, it's a bacterium, and it sets up infection by being inhaled into the lungs. And so you inhale these infectious droplets, and if somebody coughs, and that's how TB is spread. It's spread by coughing. It's not spread by touching things or sharing cutlery or crockery or by sexual intercourse, unlike HIV. It's spread really by breathing in aerosolized droplets of sputum, micro droplets of sputum, and these are inhaled into tiny air sacs in the lung where they set up infection, and about 99% of TB is acquired in that way. And the typical patient with TB, once it sets up infection, there's persistent coughing and fevers, and often one may cough up blood because TB causes literally holes in the lungs and breaks down the lungs. Well, TB is a major problem in this country. We treat about 500,000 cases of TB in South Africa, and in fact, South Africa has the dubious distinction of having the highest incidence of TB globally. If you leave out countries like Swaziland and Lesotho, which have very small populations, South Africa is #1. And it has a huge impact on society in this country. It's now officially, for those of you who don't know, the commonest cause of death, right, in this country. Surpasses trauma, surpasses violence, surpasses HIV. In fact, the commonest cause of death in HIV-infected individuals is TB. And in fact, South Africa, as many of you would know, is a single country with the highest number of HIV-infected people globally. So TB is a big problem. It's estimated that it reduces our GDP by about 5%. That's all in the region of about something just under 20 billion rand. Now, it gets a bit scary from here on, because we had drug-sensitive TB, and that's been overtaken by what we call MDR TB. Now, many of you have heard of MDR TB, and then more recently we've had XDR TB, which is highly drug-resistant TB. It's been very difficult to measure, and you may say, well, why do you want to measure infectiousness? Well, remember I told you about the super drug-resistant TB. If we can identify the small number that's spreading all these cases of TB, then we can really target these patients for isolation. Now you may say, well, what's the big problem? Just isolate all of them. Well, the problem is the numbers. To give you some economies of scale, in the United States last year, they treated 100 cases of MDR TB. Last year, we treated almost 10,000. Okay, you cannot isolate 10,000 people. And we're only treating about half the cases we diagnose. Potentially, this country has about 30,000 cases of MDR TB per year, and about 2,000 cases of XDR TB, or potentially incurable TB cases. You cannot isolate all those people, we just don't have the facilities. So all of a sudden, super drug-resistant TB has become a game changer, and it's critical that we identify these patients and isolate them, because we don't have the drugs to treat them. Why is it that diagnostics is so challenging? So one of the key impasses to TB control, there are many, but one of the key ones is to design an accurate diagnostic for TB, and we simply don't have one. Okay, and we don't have one in several ways. We don't have a very accurate diagnostic, and we don't have one where we can use this diagnostic at point of care. And what we mean by point of care is where the patient presents in the clinic. Okay, the big problem with TB is several-fold. The first thing is that there are different types of TB. The type of TB where people fall ill is called active TB, and then there's a sleeping form of TB. It's called sleeping TB, it's called latent TB infection. Now, in this form of TB, and probably about half of us in this room have the sleeping form of TB. And these are bugs that are asleep in our lungs. And if we get unwell, have a cancer, or take certain medications, or become very stressed, for example, this TB can wake up and become active. So that's the first thing. Secondly, we've co-evolved with this bug for thousands of years, okay? And in fact, there are lots of relatives of TB, and they call non-tuberculous mycobacteria. So these are cousins of TB. So it's not surprising then that if you're going to design an antibody to detect TB, it's not surprising then that such a strategy doesn't work, because there's so many people who, in fact, a third of the population or half the population in South Africa that has a sleeping form of TB, and they're well, right? We all have, half of us have sleeping TB, and if you're going to use an antibody-based approach, it's not going to work. So the conventional strategies that we use to diagnose HIV doesn't really work. And there are basically 7 things you can do to diagnose TB. One is whole pathogen detection. So you can see the bug under the microscope. As I told you, the problem with that is sensitivity. Sensitivity is only about 50%. You can grow the bug, but that takes several weeks. So that doesn't really help you, because by then some of your patients would have died. So the next major way you can do it is to detect antigen. So we do that by using an antibody-based approach. And an example of that is the LAM test. So LAM is a protein that's found in the TB cell wall, and it's actually filtered in the urine. And there's a lateral flow assay that's used to detect TB. But because of all the reasons that I mentioned, the sensitivity of that test in conventional TB is only about 10%. And in fact, in HIV-infected people, it's only as good as 50%. The third way is PCR, or nucleic acid amplification testing. And NAT, or nucleic acid amplification testing, just a fancy word for PCR, right? And basically there we amplify the DNA. Now, that's currently how we diagnose TB in the main in South Africa. And we have a new technology called GeneXpert. It's an American company. It's called Café, and the stock price has really skyrocketed because it's been rolled out. Aaron Motsoaledi likes this test. He refers to it as bazookas that will fight TB. Now, there's the major problem with this test is that you have to have a sputum sample, and I told you in about 40% of our TB patients, you simply don't have that. The second problem is the accuracy is only about 80%, right? And it's a complex technology, it's expensive, and it's not ideally suited for the clinic or point of care. There are about 4 other approaches. I'll just run through them. One is called T-cell assays, where we look at the body's immune cells, and we measure the quantity of immune cells to diagnose TB. And in fact, it's a good way to diagnose sleeping TB, but not active TB. There's also an approach that has been around for 2 decades now, called detection of volatile organic compounds. And you can actually design electronic noses to detect that. It gives you a barcode readout. It's called metabolomics, is the scientific term, and uses a mass spec approach. But when we apply that to human breath, that's where all the problems start. This idea has been around. There are companies that have been around now for about 20 years, and they haven't managed to get it right. And the reason, you may ask, why is it so challenging? Because, as I said, we co-evolved with TB. There's so many other TB-like bugs, like the question that was raised there just now. And there are many relatives of TB bugs that we have in our lungs. So that test becomes stripped up by volatile organic compounds released by so many other bugs that are being inhaled and in the lungs. And then there's metabolic detection methods where there are TB-specific enzymes that work on substrates. And that's really a new research area where people that are working on it. And finally, antibody-based assays, which haven't really worked to deliver anything for all the reasons that I mentioned, and I firmly believe that it's not going to deliver anything. So, having said that, what about the market? Is there really a big market for TB? Well, there is. The global TB market size is substantial. We have about 10 million new cases of TB every year. So the market size is roughly about 100 to 200 million tests per year. And in fact, the entire TB diagnostics market globally is now worth over $1 billion. So worldwide, globally, we spend over $1 billion a year trying to diagnose TB. And often we don't make the diagnosis. In fact, I'll give you a shocking statistic. In this country, and even in the UK and even in the United States, and I know this because I worked in the UK, I trained there doing clinical medicine and my respiratory training, only 50% of TB diagnosed in a country like the United Kingdom is actually proven, microbiologically proven, which gives you some insight into how poor the diagnostic tests are. So in half the cases, put another way, the doctors guessed that it was most likely TB, and you got 6 months of toxic drugs based on a guess. And that's really the challenge and the status quo of TB. Many of these detection platforms or technologies that I talked about are not very user-friendly, don't work very well in the field. And that's where TB is, right? TB is out there in the community. Now, in fact, we have filed a patent with UCT, and the co-principal investigator on this project is Professor Jonathan Blackburn, who's a chemist. And what we did is we looked at urine, and we got a grant from the EDCTP, it's a European-based grant, and we had a postdoc who was doing this work, and we discovered about 7 new TB-specific biomarkers. It was very exciting because we did a lot of testing to look at all these caveats, and we think we found about 5 or 6 unique biomarkers, and we've just received funding from the MRC to exploit these further. And in fact, Antrim Biotech, which is the UCT spin-off company that Andrew talked about, and the CEO is here, and you can chat to her if you want, will in fact be very closely involved in that project. It must be obvious to you now that I think this idea of developing one test that's going to solve all the TB diagnostics, I don't think that's ever going to happen, okay? Because there's so many different types of TB. There's sleeping TB, there's active TB. There's all these these atypical mycobacterial sort of bugs that are lying around. Then there's extrapulmonary TB, which is TB outside the lungs. There's TB of the lymph glands, there's TB of the gut, and they all have their own problems. A specialized subgroup of TB is TB in children. TB is a big problem in children, right? And it's very difficult to make the diagnosis. Why? Because children don't cough up sputum. It's very difficult to get samples from kids. It's very difficult to stick needles into them. So how do you get a sample? So that's a really difficult area. And our biomarkers that we discovered and that's patented at UCT, in fact, are all in urine. And in fact, urine is a very easy fluid to work with, and you can get it from kids as well as adults. So there are all these pitfalls, and so I don't think there's going to be a one size fits all. In fact, the product that Antrim is developing really is specifically for extrapulmonary TB, and that only forms about 15% of TB or 20% of TB. But in fact, in HIV-infected people, which is a big problem in our country, that extrapulmonary TB fraction increases to almost 50%. So you can see that having one diagnostic is not the answer. We need a number of different approaches, and it will depend on the type of presentation the patient has. So I'll just move on to some of the innovation-related projects that I'm co-involved with at UCT. So we've gone for a non-sputum orientated approach for the reasons that I just mentioned. So I mentioned Antrim Biotech, and they taking forward the extrapulmonary TB. There's a biomarker called interferon gamma, which is very good for extrapulmonary TB, and they're using aptamers together with nanoparticles and some proprietary detection platforms, and that's currently a project that's being funded by the IDC. We've also just been funded by MRC for a potentially disruptive technology where we're designing, making a lateral flow assay that's ultra sensitive, using several elements that I unfortunately cannot tell you about now. So I thank you for your attention and the lovely questions. Thank you.